Hello, everyone. My name is Joel Melby, and I'm a life sciences data analyst at Tetra Science. And today I'll be telling you about our integration with Benchling. Scientists use their ELNs, such as a Benchling notebook, to design and record their experiments. And then scientists execute these experiments using various lab instrumentation or automation. However, to get from a notebook to an instrument, usually there's a manual transcription step where a scientist has to type these sample names or methods into the instrument control software. And then to get the data back into a notebook, usually a scientist has to go in and find these files, maybe do some manual processing before the data is suitable to be uploaded into something like a Benchling notebook. However, what I'm going to tell you about today is how we can eliminate these manual steps using our Tetra data platform to facilitate the design and execution of experiments. And we've designed a Tetra Benchling connector that can extract sample and method information from a notebook. And then using our library of instrument connectors, we can send those sample and method details to various instruments where the scientists can just walk up to those instruments and select their samples and methods that are already waiting for them. And then again, using our library of integrations, we can actually extract various experimental data in a number of formats, including a database, if an instrument saves data in a database, semi-structured data, a tabular text file, or even a binary file. And all of these are parsed into our intermediate data schema, which we can then easily, again, pass back to our Tetra Benchling connector to upload those experimental details back into the notebook. And I'm actually going to show you a live demo of this. In this case, I'm going to design an experiment in a Benchling notebook, record some samples that need to be sent to a CDEX high-res instrument being controlled by a SmartLine Data Cockpit instance. And then we're automatically going to get the data back into that same notebook. All right, so now we're going to switch over to this Benchling notebook entry that I've already started. And in real life, we would probably have some additional details about these three particular cell culture experiments. However, for today's demo, I'm just going to go ahead and register 3 samples that we're going to use to get the total cell count as well as viability for these reactors that I have going. And so I'm going to go ahead and register these 3 CDEX samples. And I'm going to insert a run into this particular notebook entry. And so once this is inserted, I can go ahead and create a run and add these samples that I just selected to get the cell count as well as viability. And I can choose the device as well as the sample type and go ahead and hit create. And what's going to happen in the background using our Tetra Benchling connector is we're going to get that information about that run I just created and populate it onto the platform in this assay run ID.json file. And in this file, it's going to have some information about the type of run that it is. And so we know that this run needs to be sent to a CDEX high-res instrument through the SDC connector. We have the run ID that we can use to populate results back to that run. And then we have some information about the samples. And so once this file lands onto our platform, all of that information is going to be sent to the SDC instance controlling the CDEX high-res instrument. And so now if we go over to our SDC instrument, you can see I've already navigated to the CDEX high-res instrument, and I can just click refresh and we can see these 3 samples waiting for us. I'm going to go ahead and load these samples into the appropriate position and go ahead and start collecting some simulated data for these samples. And so what's going to happen after the data is available, our Tetra SDC connector is going to automatically detect those new measurements and upload a raw file onto the platform. At which point we're going to convert that raw file into our intermediate data schema where we've harmonized the data across a series of other cell counters. And then once that IDS, that intermediate data schema file is available, we're going to automatically push that data back into the Benchling notebook. We can actually see this happening on the platform. And first, we can see that initial pipeline where we got the measurement order from the assay run ID. You can see it was completed a couple of minutes ago. And now, as soon as the files are done, we should be seeing them in this raw to IDS conversion. Yep. And it looks like before I refresh, you can see a few files just landed. They're now being converted to the IDS JSON. And if we look in this push to Benchling, you can see that one file has already started. And so all of this should be fairly invisible to the scientists and will be set up by somebody in R&D IT. However, when the scientist suspects that the samples are ready, how they can check their data if their data is available, they should just have to refresh their notebook. And you can see that the data is already being populated into their notebook. And so we can see the total cell concentration as well as the viable cell concentration for these 3 samples, including a link to the raw data on the platform. And so just to prove that this is not demo magic, we can see that this 2.67 of the total cell concentration is actually seen here in this preview file. And we can also get some other information if the scientist so chose that wasn't captured in that particular result schema. And so if I go back to the slides, I hope I've shown you that using our platform, we can use these connectors to get data from ELNs and use our instrument library to get that data into various instruments, and then automatically parse that data into our intermediate data schemas and upload that data back into Benchling. Thank you all for watching. I hope you enjoyed. Bye.